Thank you for the CTSI committee for selecting my abstract for a video poster. In this presentation, I will talk about creating human ACE2 mouse model to study platelet phenotype during SARS-CoV-2 infection. This is my electronic poster, and I will explain individual sections in the following slides. Platelets play a crucial role in managing vascular integrity, hemostasis, and blood coagulation. Platelets express many receptors on the cell surface and get activated by various stimuli such as elevated levels of thrombin, fibrinogen, thromboxane E2, 1 factor, and other cytokines, which lead to degranulation and aggregation. Accumulating evidence support platelet interaction and internalization of pathogens, including viruses such as HIV, hepatitis C, influenza, Ebola, dengue virus, resulting in platelet hyperactivity. In line with this, autopsies of COVID-19 patients showed platelet aggregation in major organs, which was highlighted by CD61 staining. At present, there is spare evidence to support that SARS-CoV-2 can either directly activate platelets or cytokine-driven hyperinflammation indirectly activates platelets during COVID-19. Given that we asked two questions, does SARS-CoV-2 infect platelets? Second, what happens to circulating platelets during SARS-CoV-2 infection? To study this, first we infected wild-type and K18 human ACE2 mice and monitored the clinical changes. We observed that K18 human ACE2 mice drastically changed in body temperature, clinical score, body weight after 4 days post-infection. Interestingly, almost 85 to 90% of the K18 human ACE2 mice died at 7 days post-infection due to neuroinvasion. Wild-type mice were stable and no clinical changes and negligible amount of viral load, suggesting that K18 human ACE2 mice are susceptible to SARS-CoV-2 infection. To understand the distribution of viral loads after international infection, we applied multiple approaches. First, we infected the mice with fluorescently tagged recombinant SARS-CoV-2 virus and using IVIS spectrum optical imaging instrument and tracked the viral load in vivo. Interestingly, 6 days post-infection, we see viral load in lung and brain, but not in the organs. Next, using translational electron microscope, we examine the ultrastructure of the lung cross-sections. At 6 days post-infection, we see viral load in lung epithelial cells, but not in platelets or RBC. Further, we observe sporadic platelet aggregation in lungs. We also check the viral load in the lungs and other organs by immunohistochemistry. Interestingly, we find the viral load in the lungs and brain, but not in the kidneys and liver. Due to its contagious nature, we perform a biased mass spectroscopy-based proteome approach. Since almost 80 to 95% of the K18 human ACE2 mice died at 7 days post-infection due to neuroinvasion, we picked the earlier time points of 2 and 4 days post-infection for the platelet proteome study. Principal component analysis plot demonstrated that a clear difference in expression pattern among 2 days and 4 days post-infection and were clustered nicely. Overall, we mapped 4,600 proteins from our platelet samples. Volcano plots showed significantly up and down regulated proteins and phosphoproteins at 2 and 4 days post-infection. From these, we picked a significantly differentially expressed proteins at 2 and 4 days post-infection and compared the distribution of proteins. If you look at the Venn section, a high number of proteins fall on 2 days post-infection than 4 days post-infection. And these are all significant number of proteins differentially expressed at both 2 and 4 days post-infection. When we see the total heat map, we see many well-known upregulated proteins represent activation of platelets, coagulation, complement systems at 2 days post-infection, and interferon markers are up at 4 days post-infection. Notably, platelet factor 4 and CCL8 are upregulated both 2 and 4 days post-infection. Next, we performed pathway enrichment analysis using significantly differentially expressed proteins. At 2 days post-infection, we see a strong activation of complement coagulation pathways such as matrix receptor interaction, platelet activation, and phagocytosis. Interestingly, at 4 days post-infection, we see differentially expressed proteins are similar to influenza, reggae TLR pathways, which imply a dominant antiviral response at 4 days post-infection. We also examine phosphoproteins similar to proteome data. More phosphoproteins observed at 2 days post-infection compared to 4 days post-infection. Also, there were multiple phosphosites which are in line with platelet activation, such as PIN2, glycoprotein 1b, STIM1, and integrin beta 3. Other enrichment analysis of phosphoproteins revealed that a strong platelet activation at 2 and 4 days post-infection. Next, we performed protein-protein interactome network of significantly upregulated proteins at 2 days post-infection and 4 days post-infection using string database. The color-coded square boxes are individual pathways. Proteins in the boxes specific to particular pathways. Proteins around the boxes overlap with other pathways circled with respective colors. Notably, proteins upregulated at 2 days post-infection were more interconnected than 4 days post-infection. For instance, mononucleotide factor has been reported to play a critical role in pulmonary microvascular occlusion in COVID-19. In our interactome, we observed a high number of interconnectivity among mononucleotide factor, plasminogen, PF4, integrins, and MAP kinase, and CXCL2, PF4, and CXCL5. With an independent experiment, we also validated some of the proteins in lysates and plasma samples by multiplex and Western blotting. Interferon-dependent chemokines 6AL10, 6AL8, as well as procoagulant PF4 were significantly upregulated at 2 and 4 days post-infection lysates. Similarly, we observed elevated levels of CCL8, PF4, and IL6 in plasma. Next, to understand the translational relevance, we performed correlation analysis with publicly available human platelet transcriptomic data with our mouse proteome data. Range section reveals a wide range of correlation of SARS-CoV-2 infected mouse platelet proteome with COVID-19 patient platelet transcriptomic signature. Interestingly, major correlation observed at 4 days post-infection than 2 days post-infection. We hypothesized that the later time point correlation with COVID-19 patients due to symptomatic patients were already crossed 3 days post-infection at the time of sample collection. If you look at the correlation plot, interferon signature genes are proteins such as IFID M3, RSR2, IFID1, RIGI were significantly correlated at 4 days post-infection. We further confirmed the signature proteins IFID M3 and IFID1 by Western blotting. Overall, it confirms K18 human ACE2 mouse can serve as a good animal model to study COVID-19 associated coagulation and immune responses. As human lung autopsies showed positive CD61 staining, we looked at CD61 expression in our mouse model. Immunohistochemistry approach revealed that lung and kidneys from SARS-CoV-2 infected mice show significantly high presence of CD61 aggregates despite the lack of viral load in kidneys. Next, to study the expression pattern of human ACE2 on mouse platelets, we performed flow cytometry and found that K18 human ACE2 mouse platelets do not express human ACE2. To test the serum infectivity, we treated E6 cells with serum from SARS-CoV-2 infected mice for 1 hour, and the supernatants from 48-hour post-exposure were used to detect viral load by RT-PCR. If we look at the data, we did not find viral copies in the supernatant, suggesting that there are no detectable infectious viral particles in the serum after 2 and 4 days post-infection. We conclude that our in vivo proteome analysis shows no detectable direct SARS-CoV-2 infection of platelets, at least in earlier time points in K18 human ACE2 mice. SARS-CoV-2 infected K18 human ACE2 showed early platelet activation, adhesion, and degranulation markers. Complement coagulation cascade and hyperactive platelet phenotype were dominant at 2 days post-infection, and interferon signaling was dominant at 4 days post-infection. And abundant SARS-CoV-2 spike protein in the lung, but not in platelets and other organs, suggested that platelet reprogramming towards activation, degranulation, and aggregation is likely attributed to pneumonia-induced other factors such as thrombin or cytokine-driven responses rather than direct platelet infection. With that, I would like to acknowledge Dr. Bosman for mentoring, Dr. Inley for excellent supporting aspect, Dr. Maria for transmission electron microscope images, and Dr. Florian and Dr. Nick Crossland for the ABSL3 experimental support, and histology, and Professor Katya Ravid for the great scientific inputs. I also thank BU COVID-19R, CTSI, and NIH for the support. Thank you.